All right, so this is the Osmometer Lab, and for this lab we're going to be measuring the rate of osmosis under different conditions, and to do that we're going to need an instrument called an osmometer. So the osmometer is actually super simple. Osmosis is just diffusion of water across a semi-permeable membrane. So if we're going to have osmosis going on, we need a semi-permeable membrane. That's this stuff right here. This is called dialysis tubing. Actually, it looks like tape, but if you kind of massage it between your fingers, it actually opens up and becomes a tube. And then we're actually gonna take this tube and we can snip it and across the bottom to make a smaller part and up the side so we can kind of open it up. And basically now I have a sheet of this dialysis membrane, and this dialysis membrane is basically a plastic that has tiny little pores in it that will allow water to freely pass but will block the passage of large molecules such as sucrose, which is going to be the thing we're looking at today. We're going to have different concentrations of sucrose solution, which is sugar water. And with the sugar water in this membrane, the water can move across from one side to the other, but the sugar molecules are trapped on whichever side they're at already. So we're going to take this. I have kind of a prettier one that I already cut up here. And I've got my little square of dialysis membrane. Then the other part of this is going to be a thistle tube. Thistle, I'm not even sure why it's called a thistle tube. I guess it looks like a thistle. It's basically a tube with this little funnel on top. And we're going to actually use it upside down. And we're going to take this dialysis membrane, put it here. We're going to then be able to have one solution inside the tube on one side of this membrane. And then we're going to actually take this whole thing and put it in a beaker of water. So we're going to have distilled water on the outside of the membrane. We're going to actually have our sugar water inside this little funnel on the inside part of the membrane, which is going to create a situation for osmosis. We're going to have a gradient, an osmotic gradient, right? We're going to have concentration of sucrose on one side, a different concentration, in fact zero concentration, distilled water is zero concentration, and that's going to drive osmosis, movement of water across the membrane from the beaker into this little thistle tube, which is going to start going up and up and up. So we're going to be able to actually monitor the progression of the osmosis by seeing how far up the tube this sugar water goes as more and more water comes in to add into that. So I'm going to put this thing together. I again take my tube. I need to really seal this dialysis membrane really tight because I want to make sure any water that comes in is going across here and nothing leaks out. I'm going to pull it really tight like a drum head. Boom, 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 boom. And I'm going to use not one, but a few rubber bands. As this experiment proceeds, the pressures actually get pretty intense. So any weakness is going to end up with having water and stuff leak and sugar leak out where it shouldn't. So I am basically right now pulling this membrane really tight, making sure that it's right up against the lip, making sure that there's no leakage points. I'm going to take another rubber band and I'm going to put it around, oops, again. I can't overemphasize how easy it is to have this experiment fail if you do not really seal this really well. So there's two. I think this is probably enough. You could even do a third if you want. Now I need to put my sugar solution in here, and that's easy. You can just do with the pipette. Our old friend, the pipette. Here, the volume, actually, this is more just an instrument to get it into the little hole here. We can suck it up. As you put the tip of the pipette in here, you don't want to push it all the way down because you're going to basically create a vacuum, and you're actually going to not be able to have the sugar water go in because the air is going to block it. So I'm going to start filling this thing. Whoops. Do. And do. I'll probably take one more time. Do. Should probably stop singing. All right, so this is filled up, and I should mention the color is purely just for visibility. It doesn't affect osmolarity or the concentrations. It's purely just for color. The red and the blue, don't think of them as players in what is the osmotic rate going to be. It's just so you can see things. Then what we need to do is put the tube into this funnel. We want to make sure, again, the sealing, making sure there's no leaks is really important. So for this, I actually have Teflon tape, which is it's used for plumbing. If you haven't seen it before, it's incredibly useful for lots of household tasks, but it's really useful for osmometers. You basically wrap it around a bit, and you kind of pull, and it rips off. Make sure you haven't blocked the hole. And this is going to create this really nice tight seal when I kind of jam it in here. So again, in this case, we have 40% inside distilled water in our beaker. So, again, our independent variable here, what's changing? We've got, in this case, the thing that's driving osmosis is this diffusion, the concentration gradient, the concentration difference across the two sides of the membrane. Here it's 40% in the funnel, 0 because it's distilled water in the beaker. The other group where they fill with this 20% solution, they have a different concentration gradient across the membrane. And we'll see how having a different concentration gradient across the membrane affects the rate of osmosis. Now I'm sticking this in here, and that's what I want to see. So at time equals 0, you can see the water is at this level up the tube. And because that's basically my time equals 0, I want to basically tape this onto the ruler such that any amount of water that crosses the membrane and enters into the thistle tube, which is going to drive the movement of the water up the tube, will be super easy to measure because it's 0 right now, and as it goes up the tube, I can just see directly off of here. In fact, I probably want to push it over a little bit because then it's even easier to see because it's against the millimeters. So that's going to make it really easy to, again, in rate, as Tina was saying earlier, rate is going to be the distance divided by the time. So for any 10-minute segment, you can measure a rate for actually that particular 10-minute segment. You can say, so in that 10 minutes, how far did it move? And divide that by 10 minutes, and it can give you millimeters per minute. So while you guys go take a little break, I'm going to now set up the osmometer with the 20%.